Hello friends, today we are going to study the structure of chromosomes. Chromosomes, where it is found. Okay, so how exactly we see chromosomes and in which stage of the cell division? That is interphase nucleus. So the nucleus which is in the interphase stage, it will be having loose and indistinct network of nucleoprotein fibers called as chromatin. In the sense, inside the nucleus, when it is in interphase stage, we don't call it as chromosomes because it is not present as the structure of chromosome. It is present like a thin thread like fibers. That fiber is made up of nucleoproteins. We call it as chromatin. Just a thin thread like structures are seen. That is called as chromatin. So we don't call it as chromosome, we call it as chromatin. That is during the metaphase stage. Okay, cell division, there is prophase, metaphase, anaphase, telophase. Out of all this stage, in the metaphase stage, we see chromosomes. The proper structure is seen and we name it as chromosomes. Remember the point properly, in the metaphase stage. In mitotic metaphase, size of the chromosomes, size varies from one organism to another. So few examples are there. Size of the chromosomes of fungi, it will be 0.25 microns. Like that, few examples are given which is important. Plant chromosomes are larger than the animal chromosomes. If we compare plant chromosomes and animal chromosomes, plant chromosomes will be larger than the animal chromosomes. Largest chromosomes are lamp brush chromosome, which is seen in vertebrate oocyte, and polytene chromosomes, which is seen in the insects. In eukaryotes, during metaphase, all the chromosomes are rod shaped, which will be having a centromere and two chromatids, proper structure of chromosomes. They appear like a rod. In anaphase, different shapes of chromatids can be seen. In anaphase, these chromatids get separated and move to opposite poles. When they get separated, they appear in different shapes. That is V, L, J and I. While learning the types of chromosomes, according to the position of centromere, you will be learning these different shapes. These shapes are mainly based on the centromere position. Okay. Best structure of the chromosome. If you have to study the structure of the chromosome, the best structure of chromosome is studied in the metaphase stage. As I said you, in the metaphase stage itself, chromosome structure is seen properly because there is proper condensation. Chromatin contains DNA. The chromatin fibers, the thread-like fibers, it contains DNA and basic proteins which is called as histones. What happens is, in the histones, DNA will be wrapped around or coiled around the histones in eukaryotes. There will be non-histone proteins and RNA also. Let us see about now human cell. A single human cell has 2-meter long thread of DNA distributed among 46 or 23 pairs of chromosomes. Now human beings have 23 pairs of chromosomes. In that 23 pairs of chromosomes, totally 46 chromosomes are there. This DNA will be wrapped around that chromosomes. It will be around 2 meter. That is only a single human cell. Single human cell, if you take and see the DNA, it is 2 meters long. In prokaryotes, DNA will be circular or ring shaped. It doesn't have, prokaryotic cell doesn't have any nucleus around the DNA. So DNA will be naked but circular DNA, or it appears like a ring shaped DNA. Chromatid. So in structure of chromosomes, first you will be learning about chromatid. So chromatid, it is a replicated chromosome having two daughter strands joined by a centromere. Each chromatid has DNA molecule. See the diagram and you will come to know which are the chromatids now. See the labelings here. These structures, okay, these structures which is shaded in blue, they are chromatids. In the middle, they are connected by centromere. So chromosomes will be having two chromatids. In the center will be the centromere. Now you heard about the word centromere. What is this centromere? Centromere will divide these chromatids into different arms. So in the middle, centromere is present. It is dividing the chromosome into different arms. Kinetochore. If you see the centromere, sides of the centromere, you can see disc-shaped structure that is called as kinetochores. Centromere, if it is in the middle, here in green color I have marked a disc-shaped structure. So round structures you can see, they are kinetochores. Okay. Now first, when the chromosome is divided by centromere, they are called as primary constriction. Again, when the chromosome is divided, they are called as a secondary constriction. These secondary constrictions are non-staining. They doesn't take any stain. Secondary constrictions. At any point of the chromosome, it will occur. We also call it as satellite. Here you can see in the up. Okay. Again, this chromatid got divided, forming secondary constriction. They are not taking up any stain. They are colorless, because they didn't take any of the stain. Telomere. Ends of the chromosomes are called as a telomere. So these are the structure of the chromosomes. You can see chromosomes are classified according to the centromere, that is the middle point which is dividing the chromosomes into different chromatids. Okay, according to this centromere, where it is positioned, according to the position of the centromere, where it is placed, chromosomes are divided into different types according to the position of the centromere. First one is metacentric. Here you can see chromatids, okay, they are having equal size. They are looking similar, both the chromatids. So centromere is placed in the middle. Okay, chromatids are looking similar. They are having equal size. It attains during anaphase, when this chromatids get separated, it attains a V shape. Okay, it attains V shaped. This is very much important. Next, coming to submetacentric. Here what happens in submetacentric? Okay, if you carefully observe in submetacentric, chromatid size is differing. That is because centromere position is little away from the center. It is not exactly in the middle. It is little away from the center. So one arm is short, one arm is long. When during anaphase these chromatids separate, it attains L shape. Next is acrocentric. Acrocentric, you can see here centromere is nearby, near to the end point. Okay, it is much away from the center. One very short arm, one very long arm, attaining the J shape. These shapes are attained when they are separating during the anaphase. Telocentric, completely in the end point. Okay, completely in the end point, centromere is there and chromatids. So after this centromere, you cannot see any chromatids because it is in the end point, attaining I shaped during anaphase. Just to see the structure of chromosome one by one. So first chromosome, what you learn, they are having chromatids. Two chromatids are present in the chromosome. These chromatids are separated by centromere, which is positioned in the center. It can be positioned, you have learned types, it can be positioned away from the center also or in the ends also. Okay, generally it is positioned in the center, general structure which you are learning. In the centromere sides, if you see, there are disc shaped structures called as kinetochores. This centromere, okay, due to this centromere, chromatids get separated into two. They are called as primary constriction. Here constriction occurred, that is primary constriction. And you have learnt about secondary constriction, which is not taking up the stain. Anywhere in the chromosome it can occur, and they are called as satellites. Small microbodies are formed in the chromosome called as satellites. Okay, what is present inside the chromosome? That is nothing but DNA. In the black color fibers I have done, that is nothing but DNA, deoxyribonucleic acid. Okay, here you can see DNA, when it is loosely coiled, it is called as euchromatin, true chromatin. Sometimes it can be tightly packed, forming heterochromatin, but remember euchromatin is active. Have studied in the metaphase only, you can see this structure, that is chromosomes, which is important. No, now in the metaphase itself, chromosomes can be seen, their structure can be studied properly in the metaphase stage because they are highly condensed in that stage. Okay, if you see here, this is the outer part, that is a cell. Okay, this is a cell now, and cell will be having nucleus. Inside the nucleus you can see chromosomes. Okay, in the chromosome, what is packed, that's fiber like structure, they are a DNA. Okay, how DNA is present in eukaryotes? They are not present alone, they are coiled around the histones. Like if you take a chain, beaded chain, okay, if you take a pearl chain,